So when we talk about fats, the correct word that we should be using as a biological molecule is lipids. Okay, the lipids can be divided into two types. There are fats and there are oils. Fats are basically, what are fats? Fats are lipids that are solid at room temperature. Oils are lipids that are liquid at room temperature. You understand? You can Examples that we can generally come across are animal fats, and the examples that we generally come across in oils are many things. Uh, olive olive oil, or you can say canola oil, coconut oil. Okay, these are the examples. So what are lipids? How can you define lipids? Right? Say are organic molecules. Number one, organic molecules. Number two, made up of carbon, hydrogen, and oxygen. Carbon, hydrogen, and oxygen. But something very important. The question over here is, how are you going to differentiate between carbohydrates and lipids, right? Both of them are made by made up of carbon, hydrogen, and oxygen. If you can recall, in carbohydrates, we studied that in carbohydrates, hydrogen, hydrogen and oxygen. We discussed that hydrogen and oxygen are present into 2:1 ratio. Remember, with carbon, the hydrogen was present in 2:1 ratio. All right. But in lipids, the oxygen, the hydrogen, but uh, oxygen, or rather you can say hydrogen oxygen, or hydrogen oxygen proportion, or hydrogen oxygen are in much less proportion as in carbohydrates. Right? For carbohydrates, we studied that they are present in 2:1 ratio. Right? Like it's C6H12O6, or you can say it's C12H24 and O12, always in 2:1 ratio, no matter what. But for lipids, this is not the case. When you talk about lipid, an example of lipid is what? Triglyceride. Triglyceride, which is actually the beef fat that we generally are able to see in the meat. Beef fat has a formula of C57H110 and O6. You see, the proportion is much less. This is how you will be able to distinguish between, when you are given the molecular formulas, the only way to distinguish between carbohydrates and lipids is what? Is by looking at the proportion of hydrogen and oxygen. If the proportion of hydrogen and oxygen is very less, not 2:1, or any other except for 2:1, it means that it is not a carbohydrate. Is this clear? Okay. So there is, unlike carbohydrates, we saw that there is a general formula. For lipids, there isn't any general formula as such because the proportion varies. You understand? Basically, for carbohydrates, we had the general formula was CnH to NNON, right? This was our general formula for carbohydrates. Or you can also say it was CnH2ON. This is also the same, right? But we do not have any general formula for lipids. Is this making sense, Adia?
G.
All right. And are you writing this down, everything?
G.
Now, when we talk about lipid molecule, this lipid molecule is also called, this is also called a lipid molecule is also called triglyceride. All right, triglyceride. Well, a lipid molecule is actually made up of two components. This is made up of glycerol, which is a form of an alcohol, and fatty acids, which is a form of carboxylic acid. You might not be aware of these terms right now, but just write them down. Okay, glycerol and fatty acids, when they get together, by which reaction? By condensation reaction, they form gly gly triglyceride. All right, molecule. Is it clear? Generally, there are three fatty acid molecules that combine with one glycerol molecule. So this is the glycerol molecule, and there are three fatty acid chains generally drawn like this. Each of these peaks is actually representing a carbon. Okay, this is carbon, carbon, carbon. Sometimes in books it is also shown as such, just like a long. Okay, it could be shown with any type. You understand? So when a molecule is formed, you're going to get something like this. Up to you, you can draw a straight line. You can draw these. This is the most appropriate way to draw, but it's up to you. You understand? Similarly, if you're going to hydrolyze a lipid molecule, hydrolysis, I told you, hydrolysis is in a process which involves water and enzyme or hydrochloric acid. It could be hydrochloric acid or any other acid. You understand? It could be or. Keep in mind, it could be or. So when these things are mixed, the water or an enzyme is mixed with the lipid molecule, with a sample containing lipid, it breaks the lipid down into two components. What is that? A glycerol and fatty acids. You understand? The reverse of condensation. You're going to get one glycerol molecule, three fatty acid molecules. This is clear? Enzyme that is used to hydrolyze lipid is lipase. Is this clear up to now?
G.
Alright. So when you're going to talk about sources of lipids, what are the sources for fat, for lipids? Rich lipid, rich fat or rich lipid foods. You can say are butter, cheese, fatty meat, nuts, uh, castor oil, palm oil, and all the other oils, right? Is clear? You have to keep in mind there are certain fishes that do not have a lot of fat in them. They have a lot of protein in them. Sometimes you have to be careful in answering the question. We'll see. Sources of fat, do you understand? Important part. You can say that the lipids that we were able to distinguish them between two types. It was fats and oils. Fats are basically also called saturated fats, saturated lipids or fats. And oils are generally called unsaturated lipids and or fats. Generally, these are oils and all at room temperature. Why is this distinction? We need to understand that this difference is due to the fatty acid chains attached in each of the molecule, right? It's about the fatty acid chain that is there. For fats, for fats, the fatty acid chain is saturated. What does it mean? It means that none of the carbon in the chain has a double bond with other carbon. Right? Fatty acid chain, how are they? Organic compounds are joined like this, right? Hydrogen, 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 OH or something like that maybe. None of the carbon is double bond, has a double bond. This this makes the fatty acid chain straight and glyceride molecules are able to tightly stack upon each other, okay, and form a fixed shape. Like how? Like this fatty acid molecule, right? Maybe another fatty acid molecule is able to, you can say like this. You understand? They're easily able to stack over each other and become tightly and closely packed. Becoming tightly and closely packed in the particular at the particular term, what does that mean? It means what? That the substance is going to be solid, right? If the particles are tightly and closely packed, it means they are going to be solid at RTP. Is this making sense? Hmm? On the other hand, when you're going to talk about, have you written all of this earlier?
G.
On the other hand, when you talk about unsaturated fatty acids, which are oils, which are liquid, so liquid at room temperature, you find that the fatty acid chain in unsaturated The fatty acid chain has unsaturated carbons and chain. Like what? So, which means that they have double bond between carbon and carbon at intervals, or maybe once. So what happens is that the double bond curves, or you can say bends the chain. How? See, this is glycerol, and these were the normal chain, right? If there is a double bond here somewhere, let's say here, or if there's a double bond here, this will become bent and straight. This is going to bend, it bends and becomes straight. You see, there is a bend. So what happens is that, oh, generally these chains are not able to stay straight. They have bends and curls and curves. So what happens is, this is a glycerol. The chain tends to become like these. You get it? Is this making sense? So when the with the bend chains, hello? Yes, Hadi, are you there?
G.
So with the what happens is that, due to the presence of these double bonds, we are going to learn what does this means. Due to the presence of these double bonds, there are with the bent chains, or you can say with the kink kinked chains, the molecules, the lipid molecules are not able to fit tightly, tightly enough to form solids. Do you understand? So they are kind of loosely packed. If you look at the other ones, let's say these ones, so they can easily fit upon each other and become tightly packed. Do you understand? But as with the kink, you can see that they cannot closely and tightly pack together. Sorry. With the kinks, let's, so the particles have, they cannot closely pack together. Do you understand? So they have an appearance like an liquid, like a liquid. Is this clear? Is this making sense?
जी.
All right. So this is the difference between a saturated and unsaturated fat. Okay. And we say that saturated fats are better because they are liquid at room temperatures. They are better as compared to the solid fats, which sometimes also lead to cholesterol. So you can say that saturated fats are not really good for health. Why? They lead to the cholesterol formation, which can narrow, which cause the schema of blood vessels, which is narrowing. You understand? Narrowing of blood vessels, and which causes a lot more problems. We'll study them when we are going to be studying the heart diseases and everything. For now, we just keep it till here. Is it okay? The next thing that we have to go through is the next thing that we have to go through is uh, we've already gone through the sources. We have to see how the fats are useful. How are they used? What are the functions of the fat in the body? Okay. So first and foremost, functions of fat, lipids in the body. Number one, and for first and foremost, they are very important. A of source and storage of energy. They are very good source and storage of energy. Okay. We know that they can produce more energy than the same amount of glucose can, but it takes a very long while for it to produce. Glucose can easily be oxidized. Remember, this is the equation, right? This is the equation for respiration, right? Any organic compound, let's say if it was 57H110 and O6, even if it was lipid, this is glucose and this is pristine with the fat that we studied. They are, both of them are going to produce carbon dioxide and water, and both of them are going to produce energy, right? Energy that this produces, both of them are exothermic reactions. Exothermic, they are going to have a negative delta H value. They have both of them are going to give a tremendous amount of energy, but this is more exothermic, which means that it gives more energy. But why in but glucose as glucose as a source of energy is more efficient as compared to lipids, because it is able to produce a large amount of it is able to produce energy very quickly and easily. You understand? Right. Second and the most important function is that insulating, an insulating as an insulating material. Okay, underneath the skin, underneath the skin and around the organs